really like about our research is our labs are very colourful because we have lots of bright lasers bouncing off mirrors, so it's quite different from a normal wet lab. We develop tools to directly image molecules in cells undergoing biology, and by doing that we can see things that people haven't seen before, new biology, new processes, and we're particularly focused around the immune response, both the innate immune response, which is the first response to an infection, and the adaptive immune response, which comes later, and also that the other big theme is how proteins aggregate and cause neurodegenerative disease. And by developing these new methods, we can see new important details of these processes and hopefully use that to design new therapies or develop early diagnostics for some of these diseases. Our most major impact was very early on when we developed, using these types of techniques, a way of sequencing DNA very rapidly. So at the time, they'd sequence the average genome of a human, which is 3 billion bases, and that could cost $3 billion. And me and my colleague Shankar came up with an idea to speed that up by about a factor of 1 million. And so we set up a small spin-out company, and that went very well, and that's now the major way of sequencing human genomes. And that's now being used by the NHS, for example, where when you have a newborn baby, if it has a rare illness, they sequence the mother and father and the baby. In about half the cases, they can actually see what mutation the baby has, and there's often a treatment for it. So it's actually now being used in the NHS. We're working on three main things: how the innate immune system works, how the adaptive immune system works, and how proteins aggregate and cause neurodegenerative diseases such as Alzheimer's and Parkinson's. Probably the most exciting thing more recently is we've taken blood samples from patients who've got Alzheimer's or Parkinson's disease, and we've been able to directly see the individual aggregates that seem to accumulate in their blood as they develop the disease. So this is new information about what happens as you develop these diseases, which takes decades, and also has some potential as a method for early diagnosis of the disease, because we see differences in these aggregates, and we think that might enable us to detect early stage disease. And if we treat people earlier in the disease, we hope that maybe there'll be a better outcome, and we can also monitor how effective new therapies are at reducing the numbers of these aggregates. So it's quite surprising that we can actually take a sample out of a human and actually apply these very sensitive techniques and actually see these individual aggregates. Our research is quite multidisciplinary, so the idea is by working together we can solve a big problem. We collaborate quite widely with groups who are experts in the adaptive immune system, the innate immune system, and a lot of clinical neuroscientists down at Addenbrooke's. So we're also part of the Dementia Research Institute. We don't need to be experts in everything, but we need to collaborate with someone who can add the complementary expertise that we need to do our research. We took the group to Chamonix last year as a sort of team building exercise. We haven't been able to do it this year because of COVID. We have a couple of, ideally a summer group event, and then we have a group Christmas dinner. And then the group by themselves also organise various social events. I was an undergraduate at Cambridge, so I sort of had a better idea of what Cambridge was like when I came back as an academic. I'm not sure it was a culture shock. I think the problem with Cambridge, it's not clear how it actually works, and you have to figure that out. It's not written down anywhere.